before we get into the meat of the conversation is just some general questions. So if you had the chance to sort of bring back from the dead any scientist or thinker in the past and sit down and have a few beers and a conversation with them, what would you talk about?
And who would it be?
Well, I feel like you kind of have. I guess the obvious answer is Darwin, of course. I feel though, having heard about having beers with people, I think John Maynard Smith would be perhaps my choice. It's been said that the most important thing for a conference is not the the program or the out of conference excursions. Is that that Maynard Smith can attend and there's a big spacious bar. And the rest of people take care of it.
Yeah,
sounds like a super fun guy.
Take care of it itself. And I think what I would like to talk to him about in particular is the role of formal theory, mathematical theory in evolutionary biology. He was an engineer by training, and his maths is enough to scare a biologist, but not enough to impress a mathematician. We have some people who are very skilled mathematics and can do very sophisticated models. But I think that there is a kind of a tendency that there's diminishing returns to that, and kind of like some of the kind of rather intuitive models relying a lot on approximations, simulations that he represented, that school of approach to mathematical modeling. I think it would be interesting to hear his take on where kind of theoretical population genetics and other more formal parts of our field are today compared to when he was kind of coming up 30, 40, 50 years ago.
Mm-hmm. What things that are going on now do you think would most excite him?
I mean, he towards the very end of his career, he did some of the really early work on sequencing data. He probably would be quite excited by that. So I think that, of course, that a lot of things that were purely theoretical debates only a couple of decades ago about the importance of selection at the genome level, linkage recombination, all these kind of processes that were almost purely the subject of modeling. Now you can settle those arguments. And I think that's also why we don't see those really rambunctious debates in population genetics today as we used to do kind of half a century ago. It's simply because the data is there. You can go out and check, and debates tend to mature with that the worst, I think, kind of debates tend to happen in parts of the field that are theory rich but data poor. I would, of course, also would like to hear his take on the most recent reiteration of the controversy over inclusive fitness that's been taking place in the last 10 years. I mean, he had a somewhat contentious relationship with Hamilton, of course, on a personal level, but also he was kind of a kind of friendly critic of the concept. And I do talk about it a little bit in the book, that sometimes you hear these kind of echoes of the critique that he came up with about that inclusive fitness is a messy, complicated concept that's it's easy to get wrong. And often you're better off just doing models of classic population genetics, asking about if you have an allele with these kind of properties, what will happen to it in this population. So how he would view this, which again also feeds into this like role of mathematical population modeling, that so the kind of recent criticism came from people who are mathematicians by training, Martin Nowak, and so on, and which is, I think, has a lot to offer. But yeah, so those are perhaps the kind of topics I'd like to, and in general, I mean, he's as a scientist, as a writer, essayist, it seems to be a tremendously fun guy to be around.
Great.
Do you guys chat for sure?
One other warm-up question, and I'll give you, this is a bifurcating question, so you can answer either of two parts. And this is going to be non-scientific. So what's the most beautiful place you've ever been? Or if you're a cook, what's your favorite meal that you make?
Probably the most beautiful place. Iceland, it was quite, I've been to Iceland, kind of hiking in Iceland, some multi-day hike in Iceland. That was spectacular, mostly because it looked like nothing else. The kind of the colors in the landscape that, I've spent a lot of time in mountains, but they kind of, you get used to the palette. But here, things were red, they were orange, they were blue in a kind of completely different kind of color scape. So that I was very taken by that. In terms of cooking, probably like game and wild berries.
Oh, oh,
Like, yeah,
nice.
probably. My family's from northern parts of Sweden, so we eat
Yeah.
a lot of moose and reindeer.
Are there particular Swedish
something like that.
recipes that you would use for moose or reindeer?
Yeah, so I mean, you can kind of make like both of them. You can make nicely like stews with potatoes and lingonberries. It's pretty typical kind of northern Swedish dish. So
sounds great.
like that.
That's great.
That sounds pretty fantastic.
I've only ever spent a few hours in Iceland and Reykjavik in the airport, and really regretted not being able to get out to the landscape. I have to go back at some point and get out and see it all. So
it's a spectacular place.
Yeah.
A live event in Reykjavik, Art. That's my
suggestion. Okay, let's do that.
Yeah. Sold. Okay, so Arvid, let's jump into it. Arvid, thank you so much for joining us on the show today. It's great to have you on. Thanks for sharing your book, Gene's Eye View of Evolution. It's going to be the crux of the conversation today. And before we get into it, can you tell us a little bit about your background as a scientist and evolutionary biologist, and interested in the pathway that led you to writing this book?
Yeah, first of all, thank you for having me. I'm very excited to be part of this conversation. So I'm an evolutionary biologist by training. Did my PhD working on kind of population and comparative genomics. Been primarily interested in the kind of evolution of genomic conflicts and the biology of self-genetic elements. And I've been interested in how and why genomic conflicts arise, but perhaps in particular, what they can tell us about social evolution or the evolution of conflict and cooperation more generally. So how our empirical insights from studying genomic conflicts, how that they fit into our more general models of social evolution.
Okay.
And it was also that interest that led me into kind of a fascination with these more conceptual questions, questions about the units and levels of selection and kind of other questions that lie in this kind of territory just in between theoretical biology and the philosophy of biology. Mm-hmm. And in many ways, that's how I came into the Gene's Eye View. And the book itself kind of grew out of my interest in this debate over the Gene's Eye View, and in particular, the fascination that here you have a way of thinking about evolution that has had an enormous influence on both professional biologists and on lay people. And among both groups, it has amassed strong supporters and really fierce critics. And I was kind of interested in why is that? Why is it that when you walk up to one colleague at a conference and you say you're interested in the Gene's Eye View, they say, well, you know, that has been settled. That is the received view, and everyone knows that. Mm-hmm. And then you walk up to another colleague and they say, well, you know, this is proven in the 70s. It's been refuted ever since, and everyone knows that. So I kind of was interested in
who are these
and I was
everyone's?
interested in that. Yeah, why is that? Why is it that this kind of way of thinking continues to attract these kind of strong emotions? And I wasn't kind of wanted to write a book because I realized that this is a really sprawling literature that has kind of grown around this debate, and it's been played out for almost half a century, both in the technical literature in biology, but also a lot of it has taken place in philosophy journals. A lot of it has taken place in popular science books or kind of general audience magazines. So it's a really sprawling thing that can be quite hard to navigate, which I think is kind of partly why people tend to, what I think, have a rather unnuanced view on the state of affairs. And it kind of is, I was out of, so kind of combination of that fascination with the disparate views and a frustration that it was hard to navigate. This book project grew out of.
I think you answered my next question, which is why write a book as opposed to something else? But maybe it's worth articulating what that something else might have been. I mean, it seems like the other approach would be to do something empirical in the lab, do the critical sets of experiments that somehow convince one side or the other. So why go the route of a book rather than the route of experiments?
Yeah, so that's partly, I think, because I think that the Gene's Eye View occupies a rather peculiar position in theoretical biology. It's not a straightforward empirical hypothesis, so it can certainly help us come up with such that then can be verified or rejected. It's not a kind of all-encompassing general mathematical framework, so it can certainly inspire us to come up with more formal kinds of models. Instead, it is a kind of way, a way of thinking, a way of kind of looking at the world. And I think that it was from a kind of desire to figure out like when that works and when it really comes to its own as a perspective and when it doesn't, and what is it that why some people think that the sacrifices that you make for this perspective to work are worth it, and why some people think that the sacrifices you make are so unbearable for
I see. So you're saying it's a kind of a point of view or a framework for thinking rather than a hypothesis per se that's going to produce empirically testable things directly.
So I think it is a framework that is very good at coming up with certain kinds of predictions, and so it's a tremendous. So the way I like to phrase it, I think, is a tremendously powerful tool to think about biology. But like any other tool, you must understand what this tool was designed to do, and if you can understand that, what is it for? And this is why I think it's really important. I spend a lot of time in the book trying to understand where did this perspective come from? What is the intellectual milieu out of which it grew? What does it take to be the central problem that a theory of evolution must be able to explain? So I think if you can kind of have that on board, it gets easier to kind of use the strength of the perspective and kind of also be mindful of the its weaknesses.
That's a really nice distinction. Good.
Yeah, that's a useful way to think about things. I think we're going to come back to that perspective, and but I think obviously to folks that have listened to the show, Art and I are, let's say, skeptical about the gene's eye view, and we've talked about that offline a little bit. I'm interested, before we dig into the book, though, a little bit more about your background, about how you came to do this. It was one of the first statements, if not the first statement in the book, where you wrote that one of your biggest embarrassments in life is that you're such a poor naturalist. So I think I am too. I've become a poorer naturalist the longer I've been a professor, ironically. But if you were a poor naturalist, when in your life were you inspired to, sort of take that philosophical side to biology, and then, more recently get into the quantitative and genomic side? How did that trajectory proceed?
Yeah, so ironically, I've always been very fascinated by biology. Partly, I think for me, it really was evolution as a theory, and kind of its abstract nature on the one hand, but on the other, its ability to make these kind of predictions and this kind of ability to explain the life around us. So I very much more came into it with a fascination of that, rather than a particular interest in any specific kind of organisms, or because I've also always been rather poor in the lab. Like I haven't done lab work in many years, but as a student, no one wanted really to be my lab partner. So for me, I think it was like those issues that have lied at the intersection between kind of more formal theory within evolutionary biology, population genetics, and other ways of modeling evolution, and the kind of the questions that it raises. And I think for me, what made writing this book so much fun is that it reminded me what a fascinating window evolutionary biology is into so many kinds of problems. And I think the gene's eye view then has been at the center of many of our fields wrestling with these issues, where there are kind of technical questions about causality, or how you ought to model genetic interactions, or thinking about genes and phenotypes and so on, to the very notions of what, if anything, does evolutionary theory have to say about who we are as humans and our role in society and so on. So for me, I think it was much more of an interest in those kind of questions that brought me into it. That being said, I mean, this was very much kind of something I did in the evenings and weekends. My PhD was in a kind of more kind of conventional empirical work, with the kind of population and comparative genomics in plants, which was tremendously fascinating. And I think it's kind of really important to, even if you're interested in these kind of questions, to learn the craft, to get an appreciation of kind of how hard it really is to make proper inferences about it. But this book, in a way, was kind of a culmination this kind of side interest that I had for many years, since I started graduate school.
Great.
Good.
Well, we've been all three of us saying this phrase, gene's eye view. And so I think it's time to confront what that is. And maybe let's just lay it out. So could you say, for a scientist, what does that mean to talk about the gene's eye view? And perhaps we could just get into replicators and vehicles here right at the very beginning.
Yeah. So the gene's eye view of evolution is a way of thinking about biology and evolution in particular that kind of emerged in the 1960s and 1970s. And often I think you can kind of highlight two authors that really led the way in transforming or introducing this way of thinking. So the first is the American George Williams, who wrote in 1966 a book called Adaptation and Natural Selection.
One of my favorites all time.
It's an incredible book, and I think despite being so well known, it's still underrated in our field. It's used the sheer amount of insights that is crammed into that book on a whole range.
A short book,
Short book, whole range of topic, incredible, very clearly written, deeply theoretical, without a single line of equations. I think in a field that often highlights the importance of the spandrels of San Marco for improving the way we talk about adaptation. I think Adaptation, Natural Selection in George Williams is the one who really raised the bar for how to think about adaptation. But in this is a book that was written then, or was published in 1966, primarily written for professional biologists, for his colleagues, which is why it's not well known kind of outside of academic biology.
Yeah, so it's not user friendly necessarily.
No, it does. You kind of, to get the most out of it, you need a stronger background. In particular, kind of in contrast then with the second book that is important, which is the Selfish Gene by Richard Dawkins, which was published a decade later in 1976, which builds on many of the same insights, make quite a similar argument, but in a much more forceful way, and in an even more kind of lucid prose, a kind of an argument that can be followed by anyone. And these books both deal with several things, but when it comes to the gene's eye view, is that they pick up on the insight from theoretical population genetics that evolution can be described as changes in allele frequencies over time. But then they kind of take this one step further to argue that biologists are always better off thinking in terms of genes rather than in terms of organisms. And they do so by kind of combining this idea that you can describe evolution as change in allele frequencies. They combine that with a kind of form of agential thinking taken from the study of animal behavior, that you think, you put yourself in the shoes of the gene, and if kind of ask yourself, if I was a gene, what would I do? And this then leads into this crucial kind of distinction between replicators and vehicles, which is the terminology introduced by Dawkins. And here the crucial insight, or the crucial part of this argument, is that the reason why you want to think in terms of genes rather than organisms is that you can think of genes are what is being transmitted essentially intact, barring mutation and recombination, from one generation to the next, whereas organisms essentially are ephemeral in the sense that they are a unique combination of the genotype, the environment, and their interaction. So they're kind of here in one generation, but then gone in the next. And the only thing that's being transmitted are the genes. So here then, replicators are defined by whatever that is being replicated, which in practice has typically meant the nucleic acid, the genetic material as we know it, and they are kind of the stars of the show. But then they're housed in this other entity, which are known as vehicles in Dawkins' terminology, which is the entity that actually interacts with the environment, and there are kind of the conventional vehicle would be the individual organism, but in principle could be kind of a cell or on a more limited circumstances a group.
Yep.
And just kind of to end there, kind of the choice of words here, of replicator and vehicles, that the philosopher kind of made a similar point, that you can, this chief insight here of the genes is this distinction between something that's being replicated and something that houses the replicator. And this is the philosopher David Hull, who liked the word replicator but preferred the word interactor over vehicle. And here I think this reveals something kind of an interesting distinction between his views and that of Dawkins, in the sense that Hull preferred interactor because it emphasizes that organisms interact with the environment and with each other, whereas Dawkins preferred vehicle because it is a more passive term. It really
it's
puts
the thing that the genes are constructing, yeah.
Exactly.
So just another kind of terminology thing. So Dawkins uses the term selfish gene. You use the phrase gene's eye view. Would you say those are the same thing essentially, or do you make a distinction between them?
Essentially, they are the same thing. I think I take the gene's eye view to be somewhat broader. I mean, I think they don't, like George Williams did not talk about selfish genes, and I think, but I think you can reasonably say that he advocated a gene's eye view. Historically, I think also that selfish genes, I mean, is used to reflect a moment on the term selfish genes, which is both kind of brilliant and frustrating at the same time. It's a genius phrase that like etches on to your mind and doesn't seem to be able to leave us. At the same time, you use both selfish and gene in somewhat unusual ways. And I think in particular, selfish is used in not a very helpful way, as it kind of slides between slightly different meanings of the term.
Yeah,
but
it's the anthropomorphizing also, and ironically, it's like a super effective meme, right, for thinking about this sort of approach.
Yeah, and I should say kind of like the selfishness that. So and perhaps we will come to this. I'm perfectly happy with a kind of anthropomorphizing if it's done correctly.
Sure.
Or
yeah.
kind of
actually.
But I should say that, so the kind of the selfishness that the kind of sliding of the definition of selfish that I can find frustrating is that on the one hand you have this definition of selfishness that selfish genes use it to mean are trying to maximize their own representation in the next generation, and by this definition all genes are selfish by definition. But then you run into this issue of, well, what do we do about genes that are kind of cheating the system? So what we now refer to as selfish genetic elements, so meiotic drivers or transposable elements and so on. What should we call those? And as a field went through for a while this kind of awkward naming things. So we call them parasitic DNA, or you call them ultra selfish genes, or and so on.
Even more selfish.
Hyper selfish, extremely ridiculously over the top selfish. This is great, Arvid. I think we want to walk through the history of these ideas, and then Art and I have talked about topics like this with so many different guests that we want to devote a lot of time to that. Can I get you to, just like you did with the selfish gene versus gene's eye, what's the relationship between gene's eye view and genetic determinism? You don't use that genetic determinism term very often in your book, but it is something that is common parlance in biology now. So how are you thinking about those two?
Yeah, so I think the debate around the selfish gene very much reached its peak in the 1980s, which kind of coincided with a general debate about the role of genes in human traits and human behavior. I think I've always taken that the gene's eye view is not more committed to genetic determinism than any other branch of population genetics or most of evolutionary theory. The gene's eye view is interested in, and I think the gene's eye view is interested in the kind of question of understanding when certain kinds of genes, so certain alleles, can spread in a population. And it has often been interested in more logical questions than perhaps working out the exact mechanism of a particular trait. So early on in the book, I make the distinction between that in a way, you can think of that there are two broad ways, two schools of thought when it comes to gene-centric view, and you can kind of distinguish between a kind of logical and a literal tradition. That the logical tradition that's interested in kind of working out the logic of natural selection. How on earth can a trait like this ever evolve? And kind of typical concerns have been things like worker sterility in social insects, or traits that are harmful to the individual but beneficial to others. How can this work out? And the gene-centric view works really quite well here, because you kind of think through scenarios where this can come up. And people who are trained in this tradition tend to be quite comfortable with thought experiments and this kind of like anthropomorphizing. If I was a gene, I would do so and so. And this clashes then with kind of what we can thought of as more literal tradition, perhaps from the more mechanistic corners of biology, a lot of molecular biology, which are more concerned about kind of carefully describing the interaction between different parts of a system and really working out the kind of the causal links between them. Now, these two traditions then come into clash a little bit when you raise the issue of genetic determinism, which is a kind of an issue about how important are genes in determining or affecting this trait, which largely is a question about is a question of about development, is a question about previous selective environments and so on. And I think the gene-centric view isn't traditionally not being particularly concerned about that. They're quite comfortable talking about a gene 4, but does so in a kind of shorthand way. And in many ways quite comfortable black boxing how that actually works. A kind of a tendency that is, I think, largely inherited from both population genetics, but also behavioral ecology, which is quite comfortable thinking that this is a trait that's heritable, but how that actually works, we're quite comfortable with. So, the gene-centric view is not particularly committed to a form of genetic determinism in any more than most parts of biology, I would say.
Great. Okay. And that's a perfect segue to the next question that I have. We're going to do some more, history of biology. You talk a lot about R.A. Fisher in the book, and I think his way, pre-Dawkins, pre-Williams, his way of articulating the roles of genes in biology, and especially adaptation, you think is really important. It's a prominent part of the book, and I think it's a good place to maybe bring that up here. So you quote him, or you wrote about him, that he considered adaptation to be the defining feature of the living world. What did he contribute to gene's eye thinking?
Fisher. So Fisher, I think, probably is the one in that generation, the person within the modern synthesis generation that contributed the most to the gene-centric view. You can, in a way, say that a gene-centric view is implicit in most of population genetics, because you're concerned about changes in allele frequencies. But I argue in the book, that there's a strong case to be made that Fisher's way of doing population genetics stands out, even compared to, say, Sewell Wright or Jabez Haldane. And in particular, I highlight Fisher's, when Fisher introduced the concept of variance in 1918, he does so by kind of introducing what can be thought of as this kind of expanded notion of what can be thought of as the environment. And he constructs these models that he's interested in what are the effect on a trait when you kind of replace one allele with another. So you consider a simple diploid organism that has two alleles, and you replace one of them with another. What is the effect on a trait? And he wants to do his calculations. He wants to see, kind of hold the rest of the environment constant in order to make this comparison in a fair way. And here kind of comes Fisher's kind of crucial contribution then, is that he argues that from a gene-centric view, though he doesn't use this term, but essentially that from the allele, what's the environment is not only the biotic environment around it, the pH of the soil or the temperature in which the
external environment,
exactly. But it's also the rest of the genome in which this allele finds itself, as well as all the other alleles segregating in the gene pool, and that this also ought to be considered in the environment then. And this is a view of the environment then that comes up clearly both in Williams and in Dawkins, that they're interested in how an allele then can spread given this environment that it finds itself. And that
so not only do So not only do alleles have to interact well with the external environment, they have to interact well with the rest of the genome that's carried along with them if they're going to find success. Right?
Exactly. Yeah. And this kind of comes up in a lot of cases where kind of common criticism of the gene-side view is how it handles interactions between genes.
So how does it deal with epistasis? Yeah,
exactly. Yeah. And the answer often lies then from proponents of the gene-side view is that the environments that biologists are quite comfortable thinking about genotype environment interactions in general, and that the gene-side view is kind of take it one step further. That it also would include the rest of the genome and the gene pool. Mm-hmm. So Fisher then has this way of approaching the way he did his models, and this really comes through then in the gene-side view, that it boils down to the kind of thinking in terms of the average effects of these alleles.
So this is maybe a little bit tangential, but Fisher also wrote something else that is an ongoing mystery for me. So that's the rate of increase in fitness of any organism at any time is equal to its genetic variance in fitness at that time. Can you just tell me what that means? I've never understood that. What does it mean for an individual to have variation in fitness?
Yeah, so what you just read is what was become known as Fisher's fundamental theorem of natural selection, which must be one of the most misunderstood statements in the history,
including by me.
Biology.
I'll sign up for that too.
Yeah.
No, I mean, I think, and there is an ongoing debate still whether it's a important insight or is it kind of trivial truism. Part of the problem is Fisher's own writing. I think that sounds like one of the first times he writes it out, where what does it even mean to have variance in an individual? It doesn't make sense. Mm-hmm. He has improved that in later writings, where he emphasizes there's a rate of change in the population, and that I might well get this wrong now, but I think a modern version of the statement is that the rate of change that is ascribable to natural selection will be due to the added genetic variance in that trait.
Okay. So that makes more sense. So it's a way of saying that the rate of evolution given a particular selective pressure depends, in a sense, on the genetic variation available to it, or the heritability of that trait, which is maybe a more familiar way of saying the same thing.
Yeah, it is certainly kind of in that class of statements. Okay. Now, of course, Fisher had a very high view of the fundamental theorem, partly indicated by the name and also
the fundamental theorem.
Yeah.
All right, great. Let's see. So I think another concept, a sort of historical thing we'd like to touch on just for a minute, is the idea of inclusive fitness and Hamilton's rule, which you spend some significant time on in the book. So can you just explain for our listeners what is inclusive fitness and what did Hamilton contribute to that idea?
So Hamilton started thinking about the evolution of social behavior already as a graduate student in the 1960s, and he was concerned about the kind of classic problem of how traits that are costly to the individual who are performing them, but beneficial to others in the population. How can that evolve? And he made fundamental contributions to this, and particularly what is now known as inclusive fitness, which is his term, but also in kind of the related concept of Hamilton's rule and kin selection, which are not terms that he came up with, but are closely related to his work. So inclusive fitness, he defined as a property of an individual. So this the fitness of an individual to which you take the fitness of an individual to which you augment the fitness of other individuals that our focal individuals is interacting with, and that our focal individual has some causal responsibility for their increase in fitness or their reproduction. You add that to our focal individual, and then you strip that. From that, you then have to subtract the fitness of our focal individual, that is the kind of causal responsibility of others in the population. So kind of in a way, it's kind of the inclusive fitness is a sum of your direct fitness so the number of organisms you have, plus your indirect fitness, so the fitness of other individuals that you're interacting with, that you have some causal responsibility for. And then you have to do this subtraction step at the end to get rid of the double counting of reproductive success. Now, why? So Hamilton thought, again, highly of this concept, and it has been tremendously helpful in many parts of the study of evolution. In particular, it's been kind of thought of that it can be a property of an individual, that an individual organism can be thought of as trying to maximize, that we can think of individuals trying to maximize their inclusive fitness. And this is can be very helpful then in a wide range of questions in social evolution and behavioral ecology. And the way that it relates then to kin selection in Hamilton's rule is that you can then use this concept of inclusive fitness to make sense of traits that are costly to individuals but beneficial to others, by thinking that they will be helpful if these interactions are primarily with relatives, because the way that this kind of augmenting and subtracting, the augmenting is done is that you scale for the relatedness of those individuals. Mm-hmm. And this is kind of that classic quip from Haldane that predates Hamilton, to save your life for two brothers raised cousins, which highlights the importance of genealogical relatedness. And this is often the idea of kin selection, that you have selection among relatives.
Right. So a question, Arvid, to try to connect what we were talking about with Fisher and with Hamilton. And I mean, maybe it's just a convenience, but it would be useful for me if you articulated it. You are making all the arguments in the summaries about Hamilton's role in inclusive fitness, kin selection, emphasizing individual. But we started just a minute ago with a sort of elegance of Fisher's ideas about alleles holding environment sort of constant. Connect those dots for me.
So, I mean, Hamilton was one of the most avid supporters of Fisher probably there ever was. He was extremely taken by Fisher's arguments. Mm-hmm. So what Hamilton wanted to do is kind of develop, in a way, kind of an extension of Fisher's thinking. But what he wanted to do is to have a theory about the individual organism. And you can ask yourself, well, why would you want to have a theory about the individual organism? Well, that partly kind of goes back to this idea that organisms, in many ways, are what stands out in the living world. Organisms are kind of the ones who are carriers of adaptations. They are the entity that appears almost as if designed.
The intuitive unit.
And we're organisms too. So maybe we're a little biased in how we perceive like to be organized. But anyway.
So what Hamilton wanted to do is to have a theory to explain to explain adaptation, and he wanted to explain in terms of the individual organism. And this, in a way, then kind of leads into some of the interesting tension between a gene-side view as a concept and inclusive fitness. Because on the one hand, they are intimately linked, largely because of the individuals involved. To my knowledge, the very first time the term selfish genes appears anywhere are in the lecture notes that a young Richard Dawkins prepared when he had been tasked to sub for his PhD advisor, who was away, mm-hmm. and he was tasked with delivering lectures on animal behavior. And this was soon after Bill Hamilton's work on inclusive fitness had been published. And if you read the 64 two-part paper, it is quite hard going. So Dawkins was young and nervous, so he typed up his notes. And in order then to explain Hamilton's idea of inclusive fitness, he made the point that you can almost think of genes as they will appear as if they are selfish. Mm-hmm. So inclusive fitness is a theory about individuals in many ways. And it's a theory that reached kind of a popular audience through the writings of Dawkins, who really wanted to talk about replicators or genes rather mm-hmm. than organisms. And mm-hmm. he's always emphasized in a way that you can think of them as two sides of the same coin, or two sides of a nectar cube, that on the one, like at the gene level, you expect them to try to maximize their own survival to the next generation, and an individual organism, you expect them to try to maximize their inclusive fitness, and that they will be, it's just two ways of saying the same thing. Mm-hmm. At the same time, if you kind of, you read Dawkins' writings all the way from days up until recently in his autobiography, you will see this kind of frustration he has with the concept of inclusive fitness, which he considers to be kind of this last-ditch attempt to save the individual, as a central unit of explanation in biology, rather than kind of fully going down to the level of the gene.
Okay.
Can I articulate what I see maybe as a conflict here? And that is that, Hamilton and Hamilton's rules is about, and inclusive fitness is about relationships among individuals, right? But the gene's eye view is talking about relationships among alleles. And so you could say that, from an organismal point of view, yeah, I should lay down my life for eight first cousins or whatever. But the collections of alleles in me are related in different ways to all of these potential relatives, right? So some alleles will find identical copies of themselves in some of my relatives, but not in others. And so it seems like there's a conflict between the sort of personal organismal level and the gene's eye view. And the gene's eye view is complicated, right? Because I have many alleles, and how can they all assess the relationships among all of those potential other organisms? They can't, right? They have to do it at sort of the organismal level. So how do you resolve that conflict?
Yes, I think that is absolutely correct. But there are one exception. So I mentioned at the beginning that Hamilton did not coin the term kin selection. That's a term that was coined by John Maynard Smith in a kind of a short essay for Nature, where he contrasted that idea with the idea of group selection. Hamilton always preferred the notion of inclusive fitness, because if you think of it, as you say, most of the time, the kind of relatedness between individuals will kind of, if you consider it at a genic level, it will be the same. But that is, of course, just on average, that we share, say, 50% with a sibling, or 1/8 with a first cousin. But exactly as you point out, for an individual at an individual locus, that may well be different. And this then leads to the idea of so-called greenbeard genes. So greenbeards is a term that was coined by Dawkins to describe a thought experiment that was first developed by Hamilton to illustrate that this exact point, that the genome-wide relatedness is not necessarily the same as the relatedness as specific locus.
Mm-hmm.
Exactly. So the thought experiment works like this. Imagine a gene or a set of tightly linked genes that does three things. One, it gives the individual a green beard. Second of all, gives the individual the ability to recognize other individuals with a green beard. And then thirdly, to behave nepotistically towards those individuals. Now, you can use Hamilton's work, or inclusive fitness and Hamilton's rule, to work out that this kind of behavior, you can still get the evolution of, say, altruism with a kind of green beard mechanism that does not require it to be the individuals interacting to be genealogical kin. They only need to be related at that locus. Because what matters is not necessarily the genome-wide relatedness, but the relatedness at the locus responsible for the social behavior. Now, this was long thought to be kind of too extravagant of a thought experiment, make the point well, and it kind of really brings it home. But how on earth could you get that kind of complex phenotype from such a simple genetic architecture? We now know that there are some examples, many of them in social microbes, primarily, where you have kind of cooperative behaviors that are seen to be governed by that kind of genetics rather than kind of
quorum sensing, that kind of thing.
So perhaps my favorite example comes from beer brewing. That when you
my favorite example too. Oh wait, something else there.
Context, not the same thing, Art.
So one, yeast has been studied in the lab for a very long time, but it's been studied even longer by people making beers, professionally or otherwise. And one thing that you often have to do when you make beers is that you have to get rid of excess yeast towards the end, and you have the yeast kind of coming together in these flocks or clumps. They aggregate together. So this is kind of what is known as a flocking behavior. It's a form of kind of protective behavior that the yeast strains come together in these clumps, where individuals are at the very center or the core of the clump, are kind of physically protected from a hostile environment, for example, high alcohol content. And this seems that, so you can ask yourself, well, which individual yeast cells will come together to form these clumps? And it seems that at least sometimes this is governed by this kind of green-bearded mechanism. So if you mix strains of different species or different kind of genetically divergent strains, the ones that come together are the ones that share this one flocking.
Interesting. They sort of cooperate to flock and fall out or something.
Exactly.
Yeah,
So, so for example, if you put them kind of in high alcohol content.
Okay, so here's my issue with, and I think all the greenbeard stuff that you just talked about and that's in the book is really interesting, and I love the thought experiment.
I love the green beard. I mean, that's just such an arbitrarily and cool trait.
But if I had to think about, the vast majority of my genes and my alleles, they can't possibly be green beards, right? So like, what are the important alleles in my body, right? There's got to be things like LDH and, apolipoprotein and all of these things that can't possibly be signaling to one another. And so although I understand the sort of intellectual appeal of the green beard argument, it feels like it can't possibly apply to most loci and most alleles.
That is absolutely correct. We've gone from a situation where we thought there were 0 examples of it to knowing that there are some. So they're not non-existent, but the absolute, it's a tiny fraction of the genes that behave like this, and it explains a very small fraction of all cooperative behavior. So I would say, the first approximation, genome-wide relatedness is the or another kind of assortment mechanism is the chief.
Okay, but then if that's the case, then is it a good idea to go to this granular level of thinking about relationships among alleles in relatives as the driver of inclusive fitness? I guess another way of asking that is, do these alleles have to recognize each other in a greenbeard sort of way, or can there be inclusive fitness without that is traceable to the allelic level?
So there are two. Okay, so I think you can tease out at least two things here. So first, do you need to have, can kin selection work without kin recognition? I guess perhaps is another way of phrasing the first.
Yeah, I imagine the answer is yes.
The answer is yes. You need some sort of assortment mechanism, and they can work as long as they interact more with each other.
Yeah.
That you can get it to work. Second, is it useful to think about genes? I would say a lot of time, no. And inclusive fitness is good that it's thinking about what should you expect the individual organism to do in specific situations. And I do think that inclusive fitness it is genic in the sense that it emphasizes genetic relatedness easily. That comes easily to it. But it's not necessarily a way of thinking about what specific genes are involved or doing things. So instead, it's about thinking about what individuals do. And so I think my impression is that a lot of time you can, if you study a group of interacting individuals in the field, you can think of the rule of thumb you can use, or the heuristic you can use is that so that you can expect the individual trying to maximize their inclusive fitness. What will then kind of the consequences in this specific scenario? And you kind of completely ignore the genetics at hand, but we think about kind of like between individual relatedness and so on.
Well, good. So I really like the Greenbeard section of the book. In fact, this section where you talk about two of the other sort of concepts that have really been a point of focus of gene's eye view scientists, if we want to build silos and categories and that type of thing, which maybe we shouldn't. But in this section of the book, you talk a lot about one of Dawkins' other book, The Extended Phenotype, and just the phenomenon of extended phenotypes, and then also selfish genetic elements. So can we touch on those both? Do you have a preference of which order? It seems like this transpose on selfish genetic elements may follow more naturally from greenbeards, but what do you prefer?
Let's start with that. And it's also the part of biology that's been closest to my heart. Is kind of for a long time been my bread and butter to try and understand the kind of weird and wonderful world of genomic conflicts and selfish genetic elements. Now, it had the kind of empirical study of genomic conflict and selfish genetic elements, and we emphasize here we use selfish genetic elements to mean genes that have the ability to promote their own transmission from parent to offspring, even if it comes at the expense of the fitness of, or it has no positive effects on the individual that carries it. It can even have a negative effect on the fitness. Right. And you can kind of trace back that the observation that not all genes are, say, inherited 15 to 50% of progeny, that they seem to occur in in 1995, when it's 9% of them, that has been recognized for over a century. The first kind of recognition that this perhaps may be described as parasitic or selfish goes back to before both Williams and Dawkins.
Mm-hmm.
But I would say that the introduction of kind of the vocabulary of the selfish gene and these kind of competing replicators, and that organism as this mere vehicles or survival machines, this kind of emphasis on kind of almost like the social life of replicators, I think really helped kind of stimulate the interest in thinking about these kind of phenomena and provided a framework to make sense of them. And we now know that there are a lot of this kind of really diverse set of examples of genomic conflict and selfish genetic elements. And I think the genes of use is this is really kind of where it really comes into its own, and to think of it. A lot of these phenomena cannot really be made sense of strictly from an individual organism point of view, that you need alternative frameworks. The genes of use, I think, is a really powerful way, a really helpful way to think about it. It's of course not the only one. Some people prefer more like multi-level selection approaches, or thinking about another approach, which I think can be helpful too. But in my experience, here's really where kind of and genes of use comes naturally to researchers in the field, even if they don't really have a horse in the race about the larger conceptual issue.
Can we talk a little bit about this? One of my favorite sort of statistics and just quirks of biology: 50% of the human genome and 80% of the corn genome is comprised of transposons. So these are famously, the jumping genes that Barbara McClintock got excited about. Connect all of that for us, Arvid. I mean, how did the genomes of these two organisms, and probably a lot of organisms, come to be comprised of all of this stuff? What's a transposon?
Yeah, I mean, that is the incredible question, right? And very much that was the question at the heart of my PhD thesis, is observation that some genomes are completely littered with transposons, and they have really large genomes. And then you have other species, often quite closely related, where the transposons are much rarer and the genes are much smaller. So what on earth is going on here? So I think transposons are, as you say, these self-replicating genes. They have the ability to, in one way or another, make copies of themselves and then insert into new locations in the genome. So this is a way then they kind of increase in copy number within within a genome. But we often call them selfish because as they move around, jump, as we say, they land in the middle of other genes or close to other genes. So they kind of disrupt the expression of those genes.
And the extra bizarre element here, I just want to, for people that aren't familiar with these, the alleles and sort of changes that we've been talking about, allele frequencies in other contexts, it has to happen across generations predominantly. The amazing thing about these transposons is that even within generations, you can get the bouncing of these sequences, throughout the genome as cells are undergoing mitosis. So you're getting massive overrepresentation, huge changes in the composition of genomes within generations as well as across generations.
Exactly. So, and within an organism, so if this happens in somatic cells, then this can lead to. So in humans, it's been linked to various diseases, including cancers that can be caused by. Because on some level, this is a form of mutation. But it's also then that if you think of this as a form of mutation, in a way, this leads into why can they be so common sometimes? And part of the answer, and I don't think we have a good answer to really explain at a detailed level why it varies. We know some of the key parameters, and part of it is that what can be thought of as the distribution of the fitness effects. So if you take all the transposons, say in a genome, or that are segregated in a population, and you kind of tally it up and say, like, how many of them have zero impact on fitness, and how many of them have like a really negative one, or some of them probably also going to have a positive one. Some of them are going to end up being under positive selection. If you tally that up, I think you will find in a lot of organisms, most of the time, transposons are neutral or slightly deleterious, which means that they kind of a really important factor that's going to determine govern their frequency is drift. So you can kind of get quite far if you do this kind of large scale phylogenetic comparisons, just by considering things like effective population size or In plants, you consider things like mating system, whether they are self-fertilizing or whether they are outcrossing. So that's going to be part of the answer. And this is kind of consistent with that. The really large genomes are found not in bacteria and other things with huge effective population sizes, and tend to really streamline genomes. But then of course there's going to be many other interactive factors as well, and I think it is still an open question, and just kind of add another layer that not only does copy number of transposons varies a lot between species, so does what kind of transposons. So you can kind of divide up transposons into different kinds depending on how they transpose. So both the human genome and say the mace genome is full of transposons, but the kind of transposons that they have are quite different.
Yeah, see, this is one of those areas of these points of interface between different separate magisteria, to use one of Gould's terms, not meant in this context at all, but similar, really. This is one that's always been really interesting to me, because you're making the point that exactly where these selfish genetic elements insert in the genome, because of the functionality that they have that manifests in the phenotype, it's completely different kinds of outcomes. So to continue on that thread really, really briefly, we talked about the distribution of fitness effects among different transposons. If lots of them have no fitness effects and some of them have many, I mean, whenever we take that snapshot is arbitrary and evolutionary time, there's going to have been selection to get rid of those sorts of things, right? If there's strong negative fitness effects, a lot of that's going to go away. Maybe that's why a lot of these are negative. But the point here is this is a real point of interface between the mechanics, the details by which phenotypic variation comes to be, and the ideas of selfish genes.
Yeah. So and just to build on that, one of my kind of favorite examples, I think, in the style of genomic conflict, that really highlights both the strength and the weakness of the gene side view, is the example of cytoplasmic male sterility in plants. So most flowering plants are hermaphroditic, so they can reproduce both through male and female reproduction, so by producing pollen or producing ovules. Now, in flowering plants, you have your genetic material in 3 locations: in the nucleus, which is where half of it is biparentally inherited, half from mom, half from dad. But then you have 2 organellar genomes, typically, the mitochondrial genome and the chloroplast genome, both which are typically, most of the time, maternally inherited, so only transmitted through the mother. Now, so if you adopt a gene side view here, so if you put yourself in the shoes of a mitochondrial gene, you're strictly maternally inherited. So if you're in a hermaphrodite, any pollen that is being produced is essentially, from your point of view, a complete waste of resources, regardless of how successful the individual hermaphrodite that you are in is at siring offspring through pollen. It doesn't improve your chances of being transmitted the slightest. So from your point of view, you would rather have this individual invest all his resources into female reproduction, into production of ovules. So you can imagine, imagine then that you have a gene or an allele that can knock out pollen production, turning this individual essentially into a female, now investing all his resources. That would be really beneficial from mitochondrial point of view. And that is exactly what we see. There's several examples of mitochondrial genes in flowering plants that does this. There's sort of male sterility genes, so they can prevent the plant from producing pollen, and which means that they're guaranteeing their own transmission, because now all reproduction is through the ovules and they are transmitted every single time. And this has led to this really fascinating thing of, on the one hand, you can study the arms of the coevolution between, on the one hand, is male sterility genes and the mitochondria, and then nuclear genes that restore male fertility, so-called restorer genes. You can do all sorts of exciting things about doing different kinds of crosses, and the way you can reveal how intimately linked these pairs tend to be. So I think this is a nice, it makes kind of perfect sense from a gene side view, that if you're maternally inherited, it's in your interest that the kind of female side reproduction is what is getting most resources invested to it. It has very little to say, however, on why there are few or no examples of chloroplast male sterility, so genes in the chloroplast that makes hermaphrodites into female. From a gene side view, there should be no difference between these situations, but there clearly is. There are loads of examples of kind of mitochondrial ones, very few or none of chloroplasts.
Mhm.
So here, these are, and kind of population genetics more generally has little to say. And I think the answer was going to lie into the difference between what these organelles are doing and kind of the cellular biology and so on.
Yeah,
What how this can be
evolutionary
achieved.
legacy and whatever it means to be a chloroplast versus a mitochondria. Yeah. Mhm.
All sorts of explanations, but it's not come from this kind of simple logic, although it's really helpful in one case. And I think it has really helped us there, but it also highlights the difficulty that it can't help us all the time.
Yeah, good. Well, so can we take just a quick break for you to check the audio? And then we want to start back over again with the concept of extended phenotype, and then go into some of these this conversation about the interface between, gene's eye view and other ways to think about evolution. But
Yeah, absolutely.
is your audio okay?
It's still running, yeah.
Okay. If
Great.
you want to just keep
So
that
is mine.
going. Good, me too. I'm gonna be gone for like 30 seconds and I'll be right back.
Good. Doing good, Arvid? Like,
Yeah.
hold on. Great.
Yeah, it's fun.
Yeah, super fun. Really, really stimulating book to read. So thanks for doing that.
Yeah, thanks for having me on.
Yeah.
I'm really glad I got the chance.
Yeah. How's the reception of the book been?
Yeah, overall, people have been seem to enjoy it.
Yeah.
I think, and I hope. I mean, in a way, it really grew out of. What I really hope it can be is kind of a starting point for having these kinds of debates and discussions about these kind of larger issues in evolutionary theory. That there's kind of, in a way, for me, this book grew out of when I grew up in Sweden, then I did undergrad in Scotland, and then I came, so Britain, gene's eye view very strong. Everyone had read Dawkins before they came to university and so on. But then I came to Canada, to Toronto for graduate school, and for the first time I was kind of surrounded by not only Canadians but all Americans, graduate students and so on. And I realized that and
they just weren't nearly as in touch with it.
No, not at all.
Yeah.
They had a very different general take on all these issues. So it was out of those kind of debates that I really got interested in this, because they became the first kind of concrete example of that. Oh, not everyone thinks about this in the same way as I do, and I wonder why that is.
So do you plan on using your book for classes? Are you going to have students read some or all of it?
Well, see, I guess the best way to do it is that you teach a class and then you write the book. So because then you can kind of workshop the ideas with students. And we a colleague of mine, Manus Patton, and I, who's at Georgetown, and I are planning to do that now. This project comes up with the paradox of the organism, this idea that you have all these opportunities for genetic conflicts and cancers and other things that can kind of erode the individual from the inside. But at the same time, most of the time the individual is fine. And as much as it pains me to admit, as a student of genomic conflicts, a lot of times you can kind of ignore them. You can just think of you can kind of just think about phenotypes and you'll be fine, right?
Yeah.
But to me that raises the on a more simple level, it raises the question why is that? Why is it that? What are the mechanisms in place? So you have this restorer genes in hermaphroditic plants that restore that suppresses conflict. You have cancer suppression mechanisms and all these kind of mechanisms that exist inside of a body to kind of keep conflicts at a minimal level, so that the integrity is not threatened. So, and
we could either dive. I mean, we could dive into this now, Marty. It's not what we had sequenced, but or we could come back to this idea, because I think this is actually quite interesting. What do you think?
I'm picking up on trying to figure out what you guys are talking about. But I missed the first part of it, so I'm not following everything.
We started talking about. Go ahead.
I was going to say, so we're working on this project, and as part of that, we are kind of running this working group that will partly teach a course on it, and then try to use that to develop a kind of an edited volume, kind of in conjunction with teaching. So kind of teaching and writing and the interaction between.
Cool. Well, let's actually put a little pin in that idea and come back to it, because I think that's quite interesting.
Well, go with whatever you want to, Art. I mean, it's just a loose guide. We don't have to do this how it's written.
Yeah. All right. So we'll just go for it. Arvid, let me see if I can re-articulate what you just said, which is that you mentioned this phrase, the paradox of the organism, by which I think you meant that if we have all of these selfish genetic elements that are looking out for their own interests inside the body, inside the genome, and that might cause lots of sort of negative effects at the organismal level. So cost to organisms to benefit individual alleles that have control over some resource or some mode of transmission. Then how do we get good working organisms? And that seems actually to be a really kind of crux issue here that we should discuss, right? Because to me, what I would say is that, well, that reflects that in fact genes are embedded in these complex sets of regulatory networks that emerge at different levels, at the genetic level, at the cellular level, at the whole organismal level, and at all of those levels there are, important systems that are acting on the organism and that are making decisions, if you will, about what the organism should do and how its physiology should run. And so why not focus on that sort of integrated level of a system as the important unit rather than the genetic level that we've been talking about?
Yeah, so first of all, I think, you articulate the so-called paradox of our organism perfectly. Why, given all these opportunities for conflicts within, whether in forms of genetic conflicts or the spread of cancer lineages and so on, why is it that individual organisms most of the time function as these kind of cohesive entities that you can almost ignore what goes on the inside, and you'll be fine if you're interested in as far as phenotypic evolution is concerned.
Yeah.
So that is the paradox, and I've kind of become interested in for two reasons. So one is that when you study selfish genetic elements, you often learn also that on the one hand you have these genes that have the ability to promote their own transmission at the expense of other genes or of organismal fitness. But then you also have other genes that have often evolved the ability to suppress the spread of these selfish genetic elements, whether you have genes that can silence the expression of transposons, where you have the restorers of male fertility in the case of cytoplasmic male sterility. And of course, you also have various mechanisms to suppress, for example, the spread of or proliferation of cancers within in multicellular organisms. So on the one hand, I've been fascinated about that, this kind of molecular mechanism of that. But second of all, it goes back to this, what in a way was probably one of my most kind of transformative insights.
Oh, can you hear him?
Lost the sound.
I can't hear you suddenly, Arvid.
I don't know what happened.
No.
That's weird. It just dropped. I don't know if you can hear us, Arvid, but we just lost you.
You can hear us?
That's strange.
Oh, it's back.
You're back.
Okay, now you're back.
Oh, maybe it was the internet connection or something.
Okay, great.
Don't know.
Sorry. Okay, I'll start over. Right, so when I learned about the major transitions, the idea of the kind of that the history of life can be characterized by these major transitions, and particularly kind of known as major transitions in individuality, that you have this kind of coming together of entities that were previously able to survive and reproduce on their own, can now only do so as part of a larger whole. So we think of kind of the origins of the first genome, and then within cells you have multiple genomes, like in the nuclear and the mitochondrial genomes that are kind of the origin of the eukaryotic cell, origin of multicellularity, and even kind of evolution of particularly new social groups and societies. And that you can kind of think of that in each of these transitions, you're kind of faced with the same conceptual issue of kind of what prevents selfish behavior from destroying the common good that can be achieved through cooperation, that are kind of the integrity of an individual multicellular organism is contingent on the cooperation of the constituent parts. Or as kind of Leo Bass put it in the evolution of individuality, that individuality is a derived character, that kind of the organism as we think of it today is itself a product of evolution. Mm-hmm. So here, I mean, kind of in a way then, so what you described that you have genes and cells that are now part of this really complex systems that are kind of hard to see what you can be gained from falling down at in the individual parts. And I think that's a very fair criticism of it. I think to me, the benefit of it is that you can kind of, if you think of it from this kind of transition point of view, from kind of bottom up, you think you kind of turn it into a problem of social evolution, which means that you can kind of come at it with the tools that we have developed to study conflict and cooperation at other levels, to understand it inside of how these kind of complex systems evolved in the first place, and think of it in terms of that. So actually, it's not. I think that is a very productive way of thinking. It's of course not mutually exclusive in any way from other approaches, but I think that's kind of why I think the paradox is such a.
And just to be sure, just to be sure I understand what you're talking about. So you're suggesting that these sort of higher level emergent phenomena of like regulatory networks and physiology are, we can think of those as like social aspects of underlying individual elements that still are the individual genes and the individual alleles.
I think that is a way of thinking that still has a lot of unrealized potential. So I think in theory, I think that ought to be a good way of thinking about it. And there are attempts to do it. I think they often tend to be rather abstract and not kind of quite light on empirical detail. But I think here is one of those cases where, kind of like the mitochondrial chloroplast conflict or lack thereof, is that you need to combine, I think, these kind of conceptual tools of social evolution with a deep understanding of the empirical system at hand. Because on some level, I do think that these kind of the gene interacting networks are, on some level, another example of a social group, and that is a productive way of approaching the problem. But I also can't point to really good examples of where that has been done in a meaningful way.
So the one other thing unless Art, you wanted to follow up, I'm gonna shift gears a little bit, I guess, in a way, and just introduce something to the conversation that's not been mentioned yet and is not touched a lot in your book. I think the problem that evolutionary biology, certain perspectives of evolutionary biology is always picked on, the origins of life. I don't know how many other guests or episodes of the show that you've listened to, but we've talked to Paul Davies and Sarah Walker and Scott Turner and many different people. And I mean, I guess Nick Lane, I don't want to leave out Nick, who really, really, emphasize those stages. I mean, they're interested in for different reasons, but how does the origins of life, and especially in sort of Scott's and Nick's perspective of the physiology of systems, interface with how you're thinking about things? Because, dealing with information, dealing with energy, these are incredibly integral processes that, I mean, I don't believe they got a ton of attention in the book. So I'm just wondering, did you make that deliberate choice, or how does that fit into your thinking, if at all? Yeah,
I'm afraid to say that partly is a reflection of my lack of knowledge of the issues of the early stages, or kind of the origins of life, or the early stages of the history of life on our planet. So that's part of it. Part of it is that it hasn't really featured much in the debates over the gene side view, and I think I have few kind of informed insights when it comes to that question. I'm afraid more than I do think at some level it becomes a social evolution problem, and perhaps this is because this is my lens on a lot of issues in general, that once you have some sort of self-replicating entities that are coming together one way or another, part of the answer there will require a solution, a kind of solution to conflict and cooperation. One
thing that you mentioned along this lines, though, is Schrodinger's book, right? What is Life, which we've talked about many times on the show. I'm not going to go through that again. I'm sure most people that are listening are well aware. But that book was written in the 40s. And so Williams and Dawkins, and I mean, many of the people that you wrote about had the opportunity to read Schrodinger. What did they think? Or if I can put that question differently, if Dawkins and or Williams and or Fisher would have been trained as physiologists, how would evolutionary theory look right now?
Yeah, that's a fun essay question.
We can save that for class.
That's a great Pops question. You need like a month to answer that one, right?
Yeah. Oh, you've got 30 seconds. Go.
Yeah, but I think it touches on something, because if you think of how they kind of what happened to biology afterwards. So I haven't come across anything to suggest that the Williams or Dawkins or Fisher read it and that they had a deep influence on them. I'm sure they did, but it's not something that comes up in their writings in any significant kind of way. You can perhaps contrast that with, I think, both Francis Crick and James Watson points to that as like being a decisive,
very influential
factor, in turning their what they became interested in going down the line. So if I were to venture, I guess the press that it had a profound influence on people who went into certain kinds of biology, but on the kind of evolutionary side, as officially conceived, I think it has had perhaps less of an influence, at least in this debate, I would say.
Yeah, nice. Wanted to just circle back for a second to Marty's question about the origin of life, and and to just articulate some of Nick Lane's views here, which have really resonated with me and Marty. And I think it reflects also this sort of broader issue of how about how he and I conceive of genes and genomes. I think in contrast to your view that you lay out in the book, and Marty and I both have a very sort of systems view that I was trying to articulate about, complex systems being important units, and many of those complex systems use this tool that we call the genome, right? So we view genes much more as tools that are, really important repositories of historical information that can be drawn on in the right moment and used at the right time. But like the sort of key thing is something about these emergent larger properties of systems. And I think Marty didn't quite say this, but we were so interested in Nick Lane's ideas about the origin of life because his idea essentially is that metabolism came first, that life arose as these sort of proto units that discovered how to use ATP and to generate ATP and then to use it to drive metabolic processes, and that those sort of early proto units later evolved nucleic acids as a way to store information more reliably. And so I think there's an alignment there in the sense of like that very process of the origin of life sort of places genes in what we view as their appropriate context of being useful tools, but not necessarily prime drivers of the process. So I don't know. What do you think about that parallel?
Yeah, so when you think of, like, say, a system that is better to think about it in terms of this kind of higher level properties, do you think, is that a pragmatic view that like it's like you could like, or sorry, do you think that like in principle we could explain it from a more reductionistic way, from kind of bottoms up, but we're better off talking about it from this way, from kind of higher level properties?
I think we're better off, and that's in part because there's so many sort of emergent properties of these higher level systems that define really important things about biology. And I know emergent properties is a frustrating concept, and a lot of people object to it. But nevertheless, I think those systems have properties that you could not predict from these lower level things called genes, even though, like, for sure, different alleles can influence the properties of those higher level networks.
Yeah, so and I'm generally really quite sympathetic to that view, because I mean, I think on the one hand, I think there's never going to be a quantum theory of baseball, right? That like,
yes, alas.
Give it time. There's a lot of money in baseball. There may very well be.
and the Atlanta Braves are going to be the ones that figure it out too. So
there you go.
Absolutely. But like nothing in baseball is like incompatible with physics, right? Like it's completely. So
right.
Maybe one day we will be, but like in the meantime, it's not a very useful way of going about it.
Yeah.
And I think. So this kind of like what the best way. I'm completely and so perhaps one thing I do think in biology is that we should be quite comfortable with having multiple perspectives at approaching the same kind of questions. So I think it's important to allow that, because depending on your perspective, different things stand out, and you're going to learn different things about it. Mm-hmm. But that being said, we also need to spend some time kind of thinking about how they hold together, how they fit together, because otherwise you run the risk of just having models that are kind of you never talk to each other, and they kind of may end up being kind of mutually incompatible with each other. But I mean, even is like the parts of evolutionary theory that I've been interested in, so take behavioral ecology, which has relied a lot on optimization theory, which is a concept that was rejected quite a long time ago in population genetics, that nothing really is ever maximized. Things are just kind of, you may reach an equilibrium more, but otherwise it's dynamics. Both of these approaches have been tremendously helpful in our study of evolution. But on some level, they are also more or less incompatible in the sense that with the assumption about how the underlying processes work. And then like, I am always of two minds about how to concerned we ought to be about that, and the kind of similar issues that we have different kinds of framework that are both helpful, that how much time should how much time is worth devoting to try to reconcile it, or is that just is better to focus on the real issues at hand, which is studying these things empirically, which is as much of a as much as I'm interested in theory, at the end of the day, I think evolutionary biology is about understanding living organisms in natural populations, and maybe that's what we should be doing.
Yeah.
So I mean, at that broad level, we are all in agreement. I mean, we would be having a conversation if we weren't. But I still am not quite sure whether I'm hearing you say that integration is possible in biology. Something you said just a minute ago made it sound like we'll do us and you do you, and we're still going to make progress. But the NSF has massively invested in integration, right, in the last decade. I mean, you know, a lot of people probably don't even like that. I personally think it's fantastic. But do you think that biology is integratable, and is there any value in doing it at all?
I definitely think it is, and I think it's worth. I do think it's worth the effort to try to integrate it. I mean, I think integrated biology is one of the terms I'm never really sure what it means.
No, nobody is.
I mean, we even some places even name departments after that, and I'm never entirely sure what.
Many now, very popular.
Yeah, like, and I like as opposed to what kind of biology. I never really understood the term. But that being said, trying to see common principles across different domains of biology, I think is hugely important, especially because biology is such a large field, relying on various really quite different kinds of methods and systems. And I think this is of course very true for biology at large. But even you take evolutionary biology, which on some level spans everything from paleontology to plant ecology, from fly genetics to ecosystem dynamics, and like you used to go to an evolution meeting, people really do quite different stuff, and there at least we ought to really try to integrate.
Well, so to what end? I mean, I'm interested in your personal perspective on. Let's pick a topic. What's the one rallying cry that all biologists could get behind? The thing that we're going to integrate. Everybody's going to sort of drop their silos and play well together, and we're all going to focus on a single thing. What would you advocate for?
Oh, so I don't think I don't know if there's meaningful to think about one single thing. So perhaps one kind of integration that I found really quite fascinating recently, or over the last few years, is this kind of issues in. So I study genomic conflicts and kind of those kind of empirical systems. What, if anything, do they have in common with people who study conflict cooperation in social insects, for example? That you can kind of see that you have kind of conceptually similar issues of that you can kind of talk about conflict suppression and selfish behavior and so on. And we use the empirical systems are very different, and we use often quite different mathematical tools to do it. When is it meaningful to try to like come up with a common vocabulary to describe these different kinds of systems, or common mathematical tools to describe these systems, or when is doing so means that we are ignoring the kind of interesting biological differences in order just to try to shoehorn it into some sort of neat framework that that's meant to apply everywhere, and all that it really amounts to is saying that this will spread when the benefits are greater than the cost, and we haven't really learned that much at the end of the day. I think so. Here, in practice, it's a narrower kind of integration.
Well, and that's understandable because, as you say, evolutionary biology is just evolutionary biology is fracture, and then we can include medicine. And biology is so ridiculously broad that integration, even within its subdisciplines, is non-trivial for sure. I honestly, though, expected you to say, and selfish, but you wrote so much about it, and most everybody else that we've been talking about has focused on it. Why not adaptation? Why not adaptation as the point of focus?
Oh, so I mean, that I can buy. I think adaptation is one of the most important issues in biology. It's clearly not everything, and whether it can be the unifying feature like that, I'm not sure. So I'm not sure exactly what that would look like. I think, like, when it comes to adaptation, I think it would be interesting to see different approaches to it being integrated. Perhaps, like, the integrations between inclusive fitness methodology, and that, like, at the end of the day, if you use inclusive fitness, you usually, what you get at the end of the day is kind of like a direction of change. Will this trait, will this spread or not in the population over time? Which quantitative genetics is historically concerned about estimating strength of selection and its response. So there, I think, like, those two both concerned with adaptation. There, I think integration would be really helpful. It would also be interesting to, like, to the extent that can be integrated with genomic approaches to the study of adaptation. Interested in kind of what are the genomic regions involved. So that kind of integration, I think, is helpful. But I'm not sure I would go so far to say. I think perhaps adaptation plays a huge role in the history of the gene side view, because it's a way that takes adaptation to be the central problem that we should try to explain. Yeah. And that's why it's often quite comfortable with thought experiments and this ignoring other kinds of details in order to understand how something like this could evolve. I'm sympathetic to that tradition, both because it's historically has been very strong, but also because I think there's very much, like, I think organisms are what makes biology unique, and one of the most defining features of organisms is adaptation. So I think adaptation deserves a very central role in anything we do. Right. But of course, there's all this other interesting things that have nothing to do with adaptations.
Sure. The quirks of what else it means to be alive on Earth. But the thing about adaptation that I think is appealing, and the reason that it is always in the background of our conversations with physicists, neuroscientists, I mean, the gamut of biologists that we talk to on the show, is that it really is about the regulation of information and energy, right? It's what distinguishes life from non-life, and the processes by which all the diversity of life came to be. It's not to say that other evolutionary and ecological, many processes don't intervene. We talked to Scott Carroll about his favorite topic about, just sort of vicariant events in the past. Chance strongly influences things. Well, yeah, of course, but still his interest in chance largely has to still do with how we have this adaptation versus that adaptation.
Yeah. No, I think evolution straddles that interesting balance that, like, Mendelian genetics and everything, make it so tantalizingly close so that you can come up with kind of simple rules about how everything ought to behave. But then at the end of the day, it's a historical science. So all sorts of things happens. Couldn't have this way.
Yeah, sorry, that was a little bit of a soapbox about things, but we went in an abstract direction, or is on an unscripted direction. I think let's try to bring it. Let's try to connect these different perspectives by juxtaposing a couple of different thinkers. And Arvid, how are you doing on time? I know we've been going for about 90 minutes now. Are you still okay?
Like 10, 15 minutes or so.
Oh. Yeah, absolutely.
Okay, good. So one of our very first guests, I think it was episode number 2, was Dennis Noble. And Dennis was on Richard Dawkins' dissertation committee, which, he told us, I had no idea until he said it. But he has a couple of really neat books that Art and I like a lot. And in one of them, I think it's Music of Life, he quotes one of Dawkins' most famous passages in Selfish Gene, which you've done. I mean, you've quoted both of these things. And so for the listeners, I'm going to read both of these. Dawkins' version is, now they, genes, swarm in huge colonies, safe inside gigantic lumbering robots, sealed off from the outside world, communicating with it by tortuous indirect routes, manipulating it by remote control. They are in you and me. They created us, body and mind, and their preservation is the ultimate rationale for our existence. Very few scientists write that way. That's pretty amazing. He has a gift with language. So then Dennis takes that text, and the physiologist that he is changes a lot of it. Now, they, genes, are trapped in huge colonies, locked inside highly intelligent beings, molded by the outside world, communicating with it by complex processes, through which, blindly as if by magic, function emerges. They are in you and me. We are the system that allows their code to be read, and their preservation is totally dependent on the joy we experience in reproducing ourselves. We are the ultimate rationale for their existence. So that's fun. I mean, it's rare to see two super impactful biologists use such poetic language to make their cases. But, you know, it seems, what is it about Dennis's sentiment that doesn't work for you? Or how do you feel about Dennis's perspective in general?
So I think the really helpful thing about putting them next to each other is that in a way it highlights the insight that I think that a gene-side view is a very helpful perspective, but because there is no way that you can kind of distinguish between these two ways of writing by coming up with some super clever experiment or like ingenious mathematical model that no one has thought of. I don't think that is what's going to settle this. I think both of these are ways of thinking that emphasize different things. So I think for me is that I take the first one, the Dawkins one, to be a more productive or helpful way to approach a lot of the questions that I've been interested in. Mm-hmm. And of course, this kind of reveals my bias in that the questions that I have been interested in are those related to social evolution, to specifically kind of study genomic conflicts and other kinds of social evolution. And there, I think that kind of way of thinking is more helpful. But so overall, it's kind of a pragmatic thing for me more than anything. What is a helpful way if we want to understand this?
Okay, fair. I mean, I take that point. I still go back to is it possible that to do integration? Yes, it's possible to do integration, and yet we have different aims. It's always going to be the case that there's details within these different magisteria that require a lot of work. I definitely agree with that. But the one piece that I always get hung up on, and Art's going to ask something about Dennis Walsh, another guest that we had on, but a different point that I'm going to raise. When we talked to Dennis, if anybody wants to go back and listen to that episode, what struck him and what has resonated with me is that when Fisher and the other architects of the modern synthesis invented statistics by all intents and purposes, and did some amazing things, that has progressed biology so far. And yet in the ensuing years, biology has come to be revealed to be a quite different thing than what biology used to be. It doesn't quite seem that the Fisherian descendants are embracing the entirety of biology. And so how we get to integration when there's not the full embrace of all the diversity of what comprises biology, I'm confused.
Yeah, so I think that there are a couple of things here. So first, of course, Fisher's descendants are only a small part of the kind of general, even if you want to call it the received view of evolution biology of population genetics. You know, Sewell Wright also had really quite different views on many things, and had a much stronger emphasis on integration between kind of much more of a systems view in a way. The emphasis on complex interactions that can't be handled by simple focus on alleles. The issue that the kind of biology today is very different from what it was then. Yes, that is very true. And whether we are doing a good enough job at integrating this into our core theories, of course, has been, I mean, ever since the term modern synthesis was coined, there's been complaints that specific areas were not given the credit that it deserves. I think that kind of evolution has proven to be in some ways, remarkably flexible in integrating things. But also there is, of course, kind of a cultural thing of what it doesn't really consider to be that important. And maybe some of these issues, insights from physiology are included there. But yeah, here I think I am probably more in the orthodox view on things that I must admit.
Understandably, I mean, the difficulty with biology is there's so much to cover. How do we educate the undergraduates and even in high school to get into your brain all of the things that are necessary to walk towards synthesis? That's a really difficult thing. The development of biology has been all about its diversity. I mean, how do you not confront that? But then to pile on, the many different conceptual ways to think to get to synthesis. Yeah.
Let me ask one more diversity question, and then we'll ask you just a couple of wrap up things here at the very end. And let me articulate a particular thing about genes, which I don't know, you may or may not agree with, but it feels to me like the primacy of genes and alleles in the gene's eye view of the world comes in part from thinking about genes and alleles as sort of the ultimate historical artifacts, right? They're the sort of oldest things that have been produced by processes in the past that are now in the present and having effects in the present. And in our talks with Dennis Walsh and with Scott Turner, they raise these ideas of sort of putting genes into the context of a broader set of what they call memory tokens. I don't know if you've run across this phrase, but they're sort of things from the past that influence the present that come in many different ways from the past. And those include things like epigenetic marks, maternal factors that moms put into eggs, ecological inheritance, so offspring inheriting things that their parents or their ancestors have built or done to the environment. So what do you think about why not just take a broader view of memory tokens and all of these influences from the past, and why give genes primacy as opposed to some of these other historical influences?
So I think there is a minor irony lurking here somewhere about that. With the concept of replicators, the important reason why Dawkins preferred that was to kind of free it from the material concerns of kind of that how inheritance happened to work here in terms of nucleic acid, that he preferred the term replicator to gene for the very reason that it emphasized that this kind of information that can get copied and not kind of in terms of kind of genes or DNA, even though that's kind of how it happens to work here. But he wanted the concept to be broader. So I think so when you want to kind of expand this notion of heredity, other things that are faithfully transmitted, there are one, I think this is an empirical issue of how often and how reliable that happens. But if we can kind of put that to the side for a moment, let's assume there are other things that are as kind of or reliably or faithfully transmitted as nucleic acid is. You cannot, there hasn't been much in terms of trying to incorporate that into kind of the replicated concept or kind of how it handles it there, because I think I think there is some potentially interesting integration to be done. But ultimately, though, I would say that kind of like how much of a concern is this kind of other forms of inheritance to a gene's eye view will depend on the empirical question of how important they actually turn out to be in the long run. Because I think even strong proponents of other forms, whether it's maternal effects or isolation marks or other forms of cultural inheritance or what form it may take, I see little evidence that it is as reliably as genetic material. But
so you think they're not as important? Is that because they're not as transmitted as faithfully as genes or alleles?
I think that will be part of it. Yeah, for sure. But I mean, I think that is an empirical question, so I'm very open to revise that. Yeah. But I think.
I see what you're saying, and I think I may agree with that at some level. Just one quick point about sort of like time decays and time over which things have influence. Like I feel like, epigenetic marks in many ways, they're transmitted almost as faithfully as genes. They just are much more flexible and changeable on shorter time scales. And so these feel like sort of different memory tokens that are tuned to different time scales of historical influence, and that actually is a key thing that gives organisms, these units, the ability to have adaptive phenotypes that work well in their current environments.
Yeah, maybe even evolvability, right? I mean,
yeah. And so I mean, I think so. I have two reactions. One, I think is like my gut reaction is perhaps the unfair one, where it feels like that might well be true, but then that is often hyped up with a lot of big implications. I think perhaps the interesting way is like also say that is the case, which may well be. To what extent can we then use our traditional tools of population genetics that model this kind of method? But it probably would be something with a very rapid back mutation rate, because like as I understand it, these kind of signals then kind of you lose them or they revert back to the original state. So can you take kind of the frameworks we have, incorporate this new empirical information and kind of still handle it? Because I mean, population genetics has been able to incorporate all of these sorts of kind of molecular details into its framework. Perhaps the best examples are models about GC bias and kind of reinterpretation, which is wasn't something that any of the architects of population genetics knew anything about, but modern science do, and we can incorporate them into those frameworks. To what extent can we do that with these kind of methylation marks? I don't know, and people are doing it already. It's not a literature that I know that well, but yeah.
Okay, great. Yeah. I don't have a wrap-up question in mind, but you have some, because I've been just thinking about this.
Arvid,
so
this has been great. I mean, thank you so much for talking to us about the book. There's two of us here that are confronting you with very difficult questions, rapid fire. It takes a special person to do that. So we really appreciate your willingness to do this and your generosity with time. I think we could probably go on for another 3 or 4 hours. We really do have so many questions. But
we'd have to do it over beers in a bar somewhere.
Let's not, because exactly, John Maynard Smith wouldn't be happy with us doing a bit in the current way. We need to have a beer if we're going to proceed. So we always wrap up with our guests. Just it's a broad question. Take it as you will, any direction. What else would you like to say? What did we not touch? What else about the book do you think is important to get across? Anything else to share?
So I think there are kind of at least two schools of thoughts when it comes to how scientists or biologists should approach their history. When I started, just before I started writing this book, a colleague of mine is in computational biology described evolutionary biology as a field obsessed with its own history. Mm-hmm. And I know that there are some colleagues of ours that kind of are more of the attitude that reading rots the mind, that like you shouldn't clog your thinking with what other people have done, because then that means you won't be able to make any original kind of contributions. I am very much the schools of thought that think knowing your history is important. So if anything, I hope that this can be kind of demonstrates if not the importance, at least the excitement of learning the history of, or modern history of the field. And I kind of hope that the book can be read with that in mind.
Excellent. Well, great. Thank you so much. We really appreciate it.
Thanks, Arvid.
Thanks for having
really
me.
Fun conversation. So yeah,
it was really fun, and as you said, hope we can do it over beers at some point.
Yeah,
we will do.